Our second speaker today from molecular genetics and microbiology is Lawton Chung. He graduated from the University of California, Irvine in 2010 with a BS in biological sciences. While at Irvine, Lawton worked in Dr. Naomi Morissette's laboratory, elucidating the function of novel cytoskeletal-associated proteins in the protozoan parasite Toxoplasma gondii. Did I get that right? Close. Okay. Following a brief stint in industry working on platforms for allergy diagnostics, Lawton joined the Department of Molecular Genetics and Microbiology as a PhD candidate in Dr. James Bliska's laboratory in 2012. His dissertation research is focused on host pathogen interactions with an emphasis on Yersinia subversion of innate immune responses. His talk is Infection by Injection, How Pathogenic Yersinia Activate and Subvert Immune Responses.
Thanks, Dr. Tabor, for the kind introduction and the opportunity to talk about my work or the work that's going on in our lab. Thank you all for coming. All right, so I want to talk to you a little bit about what we do in the lab and in regards to, and I think this is really interesting, how these tiny microorganisms or microbes can cause just very, very bad diseases. And so just a brief outline, I'll talk about the history of plague then and now, as well as Yersinia, the causative agent of plague, and the immune system. And so plague, as you all know, has been an important part of human history. And there are three major pandemics that are associated with plague. The first is the plague of Justinian, which I guess ravaged the Eastern Roman Empire and accounted for about 50 million deaths. And more famously, the Black Death, which took place in Europe between the 14th and 17th centuries, wiped out more than 200 million people. And more recently, in the 19th and 20th century, up until about 1950, there's this third pandemic, which started in Asia, and it accounted for about 12 million deaths. And so, total, that's, taking all this combined deaths, this is about 35 to 90% of the US population in 2014. So that's quite a lot of people. And so after 1950, plague actually is still around here. And so there are regions that plague is endemic to, primarily some regions of Africa, South America, and still Asia. But we also have some plague cases here every year. And to zoom in to plague in the US, each of these dots here represents a case of plague between 1970 and 2012. And you can see that it's still around. And more recently, from 2012 to 2016 or 2015, the number of plague cases or reported plague cases are on the rise. And so what are the symptoms of plague and what makes it so deadly? So the problem with plague is that if you do contract plague, and I apologize for the image, it's kind of graphic, you contract these nondescript symptoms, and they include fever, chills, swollen lymph nodes, and the important thing is that even if you catch it early, even if you diagnose plague early, you still have an 11% chance of dying or succumbing to plague, which is not very good. And however, if untreated for this form of bubonic plague, you can have 30 to 60% mortality, and that increases, and that's a pretty significant increase. And shown here is a patient that did contract plague. He survived, but they had to amputate his fingers just because there's all this death in his fingers. So actually, most people attribute plague and associate plague with rats, and that's not that's actually not entirely wrong, but it's also not entirely right. The rats are actually the rodent reservoir of plague, and they carry these fleas. And actually within the fleas, there are this bacterium, Yersinia pestis, and this is actually the causative agent of plague. So no matter how many rodents you have, how many fleas you have, if you don't have this guy right here, you're not getting plague. So actually, on the surface of these bacteria are these structures known as type 3 secretion systems. And so these structures actually extend from the bacterial surface like needles, and they contact the host cells. And so these are kind of like syringes that are found on the surface of the bugs to deliver these proteins, these bacterial proteins, directly from inside Yersinia into the host cells. And so this is how they cause disease. And one of these effectors the effector that I study in the lab is YFEM. And so it's an important virulence Yersinia virulence effector, and it's important for Yersinia pathogenesis, meaning that if you have a mutant strain of the bacteria that doesn't have YFEM, it's attenuated. And YFEM itself is a leucine-rich repeat, and once it gets delivered inside the cell, it will interact with multiple host proteins. And these interactions are important for its virulence, but not very much is understood of the consequence of these interactions. And so from this, because bacteria interact with host cells and host cells then interact with bacteria, we ask two questions. And first off is, how do host cells respond to Yersinia infection? So normally, when host cells come into contact with bacteria, there's this process called inflammation, which is a host cell response to limiting infection. However, in the presence of YFEM that Yersinia injects into the cell by its type 3 secretion system, it's able to inhibit inflammation to replicate to high numbers, eventually overwhelming your immune system. And so it's not quite known how or what pathway YFEM inhibits, just that it inhibits inflammation. And so to study this interaction between this Yersinia and host cells, we can use these very powerful genetic tools. We can use mice, mutant mice, and as well as a wild type or normal mutant Yersinia, and isolate these cells from these mice and infect them with Yersinia and just look at inflammation or lack of inflammation. So the particular pathway that we were focused on is this inflammasome pathway. And so inflammasomes are these structures that are within our cells that sense infection. And after they sense infection, they'll assemble and drive inflammation. So in a resting cell where there's actually no inflammation, like this unstruck match right here, these inflammasomes are made up of various components. And so when there's no signal, they're just floating around and they don't associate with each other, and that's kind of good for us. So, because you don't want inflammation without any infection. However, when you get a bacterial infection, viral infection, or other types of infection or injury, these parts all assemble into this inflammasome complex. And this complex will then drive inflammation. And so this is your body's way of telling or just telling itself that it's under attack. And so what we see is that, and apologize for the images, the fluorescence is not very good. So in uninfected cells, the inflammasome components are not assembled just because there's no infection. However, interestingly, when you have infection with wild-type Yersinia, which is in green, and the cells are actually in blue, if you can kind of see, you still don't have assembly of inflammasomes. So there's no assembly right here. And importantly, when you get rid of YFEM, so this is the YFEM mutant, and you infect cells, you can see that these inflammasomes start to appear, and these are little foci of inflammation. And so this is illustrated here, and this is basically what we're seeing. We're seeing this, so what we're seeing here in the red is actually this one component. We're staining specifically for this one component in the inflammasome, and when they assemble, you can actually see all of it assembling into one foci per cell. And so to look at an even more molecular view, once these guys are assembled, it leads to this cleavage of this last protein known as caspase. And we can actually have readouts and measure cleaved caspase itself, as well as the products of cleaved caspase, which are cytokines that are pro-inflammatory. And so the cytokine that I'll focus on is IL-1 beta, and we'll also measure cleaved caspase. And so this is consistent with the fluorescence data that we see in that when we infect cells with Yersinia, wild-type Yersinia, and measure host caspase, cleaved caspase, or active caspase, you don't see a band here. But when we remove YFEM from Yersinia and infect those same cells, you see that there is caspase. And so beta-actin is just a control to show that there is, in fact, protein that's been loaded here. And so we can actually read out the amount of IL-1 beta that's being produced. And we see that uninfected cells don't have IL-1 beta, don't have much IL-1 beta being released. And during infection with this wild-type Yersinia, it also inhibits inflammation. But when YFEM is absent, then inflammation goes up. So this is actually very bad for the Yersinia because they can't replicate effectively. And so your body is able to clear the infection. So hopefully by now I've shown you that this inflammasome activation will then lead to this cleavage and release of IL-1 beta from the cells. But what I didn't say is that this inflammasome is actually interchangeable. So when inflammasomes get activated, they'll all lead to caspase-1 activation and IL-1 beta secretion. But there are different components of inflammasomes that will assemble in response to different stimuli. And so the different inflammasomes are NLRP3. And so they'll sense ATP or membrane disruption. And membrane disruption happens when the type 3 secretion system is interacting with the host cell to inject the proteins. So NLRC4 recognizes flagellin or type 3 secretion rod proteins. And Pyron senses just disruption in cytoskeletal, in the cell architecture. And so these are all equal candidates for this, for inflammasome activation in response to Yersinia. And I won't show you the data, but we've excluded that these NLRP3 and NLRC4 as being important for Yersinia infection, or infection with the YFEM mutant. However, when we infect these Pyron knockouts, you can see here that once again, in the wild-type cells infected with wild-type Yersinia, when we're measuring caspase-1 activation, these Yersinia are able to inhibit caspase-1 in a YFEM-dependent manner. And so they drive caspase-1 activation. And however, when you remove Pyron, or that one particular component of the inflammasome, you lose this inflammation. And so that's better illustrated when we measure this pro-inflammatory cytokine. You can see that wild-type Yersinia are able to inhibit IL-1 beta secretion. The YFEM mutant triggers inflammation in IL-1 beta secretion. However, when you remove host Pyron, you lose this inflammation. So this indicates that Pyron itself is important for driving this inflammation in response to this YFEM mutant. And so, yeah, these are in cells, but we wanted to take it into a bigger system or a live system. And so by using these knockout mice, we can investigate whether or not Pyron is actually important for shifting the outcome of infection. And so, so what we did was we took Yersinia and we injected wild-type mice, or these Pyron knockout mice, and we monitored survival for 21 days. And I only plotted to day 15, but things don't change after that. And so on the y-axis, this is survival of the mice, so from 100 to 0, and on the x-axis is days post-infection. And so when wild-type mice, which have Pyron, are infected with Yersinia, wild-type Yersinia, it's shown in the solid line, as indicated here, they all die. They all succumb to infection by day 8. However, the YFEM mutant, just because it's lacking YFEM, triggers inflammation. And so this inflammation is really bad for Yersinia, so all the mice, or most of the mice, still survive. However, in Pyron knockouts, when you don't have this response, now it doesn't change the outcome, the infection with the wild-type Yersinia, but now this mutant is fully virulent because the host can't respond to this mutant. And so all the mice now die by day 8, the Pyron knockouts. And so to measure the actual numbers of bacteria in these mice after infection, we did the same thing, except 5 days after infection, we isolated the spleen and the liver, and then we counted the bacteria. And so on the y-axis, these are bacterial numbers, and each dot represents one mouse, and this is a log scale, so this is 10-fold lower. And so in the wild-type mice infected with the wild-type Yersinia, there's this level of colonization, and with the mutant, the YFEM mutant, is unable to colonize or is defective for colonization or in these wild-type mice because there's this inflammatory process. And you can see this both in the spleen as well as the liver. However, when you remove the response by knocking out Pyron in these mice and you do the same infection, now this mutant grows better than it did in wild-type mice, signifying that these responses in fact do kill this mutant in vivo. And we can see that this is growth is restored in the spleen as well as in the liver. And so what does this have to do with humans, plague, and Pyron and everything? So interestingly, there are mutations in Pyron. These are gain-of-function mutations. And it leads to a disease termed familial Mediterranean fever. And this is characterized by recurring episodes of fever, rash, and inflamed joints. And you can see here in this picture, this patient is having an attack where there's just a lot of inflammation here, swollen joints. And this is actually normally what it looks like. And so it's an autosomal recessive mutation, and the carrier frequency in the Mediterranean population is as high as 1 in 5. But the genetic basis or the reasons for why it's so high remain unknown. So it's speculated that the reason why these mutations are so apparent or carried to such high frequency in the population is because of some sort of infection. And so we're currently investigating whether or not plague contributed to this high carrier frequency. So in conclusion, hopefully what I've showed you is that pathogenic Yersinia use type 3 secretion system to deliver YFEM into host cells. And so inside the cells, YFEM inhibits inflammation, inhibits Pyron to restrict host inflammation. And so with the mouse experiments, what we show is that the Pyron inflammasome is important for host protection to this mutant. And so understanding these interactions between Yersinia and the immune system can provide better insight into human diseases. So with that, I want to thank everyone, especially my lab and other collaborators, and thank you for your time.